Does it work? No. Yes. Yes. Okay. So we are going to go to less complicated stories and more practical. So I left you yesterday when I was showing you that you could have a simple relationship between the curvature of the membrane and the protein concentration. So now you know well what the curvature of the membrane is. So you are now very well equipped. And so I'm going to continue on this line and show you experimentally how we can test this kind of hypothesis. So we are going to work to talk first on this kind of protein, which have a very characteristic shape. They have a banana shape, as you can see here, and they are called bar domain proteins. So they are very common on many proteins, and they have this curved shape, which could be more or less curved. In this case, it's called N bar. When it's less curved, it's called F bar, and they even can have a negative curvature. I mean, this opposite curvature to this one, and by convention, it will be called negative curvature, as you know now. And they are called inverted bar, so I bar. And this protein, they have in common that they all have a positively charged residue. So remember yesterday I tried to explain one of the reasons why we get negatively charged lipid, and one of them is because proteins tends to bind to negatively charged lipid. It could be phosphatidylserine, it could be phosphoinositol lipid, and they have this part here which is usually contains a positively charged residue. So they can bind to the membranes tightly using electrostatic. And so what happens when they bind to membrane? So if you have a flat membrane and containing negatively charged lipid, you have this protein in solution. So what has been shown with many simulations that the protein, when they bind to the membrane, they locally bend it. So you can see that the membrane is not flat anymore. And you can see on this simulation that it creates a local bending. And in the case where the protein have this bar domain plus the amphipathic helix I described just before finishing yesterday, you could also have also bending. That's the case here. And you can see that the amphipathic helix, if you look from the top, they are embedded in the membrane with again, the hydrophobic part, which is inserted between the head group, and the hydrophilic part, which is facing the outside. Okay, so and with that, they are able to deform membrane with the bar domain and the amphipathic helix. But they can also do the same thing without amphipathic helix. So I think I already showed you some images yesterday of the transformation of spherical liposomes when they are incubated with protein. And in this case, what you have here is different bar domain protein with different type of bend structure. And you can see that if you have this structure, which is not too much bent, in the case here of this protein called FBP17, you have a radius of curvature, the radius of the tubule, which is 60 nanometer. And if you go to a more curved structure, you can see that also the tube diameter is reduced. And now you have radius, in this case, of 10 nanometer. And so you can see that there is kind of a connection between the shape of the protein, whatever it means, the structure of the proteins, and the deformation of the membrane. And in the case you have a protein which has an inverted curvature, so the protein will bend the membrane in the other direction. And if you have a liposome, what you can see here that the liposome now contains kind of invagination, like tubular invagination. And you can see that they form, instead of forming tube like that, they form tube towards the inside of the vesicles. And in this case, I mean, and I will use this number later, the radius is about 20 nanometer. So you can do that also with giant liposomes. So I showed you yesterday giant liposome. Giant, remember, this about typically 10 microns. And you can see that if you incubate them with a high concentration of protein, meaning micromolar, and you have a charged lipid in the membrane, you can also get tubulation. So you get tubulation toward the outside when the proteins have a positive curvature. So these proteins, this one is also almost flat, but you see it's able to deform the membrane. And again, you see clearly that when you have a protein with the inverted curvature, you create a negative curvature and you get invagination. So that's what we have at high concentration. So in cells, the concentration of protein is usually not that high, it's not macromolar, except maybe for actin, but usually the concentration of protein is small, especially of the bar domain. They are more at lower than 100 nanomolar. And these proteins are found in many places in cells. In many places where you have some band membrane, you are sure, if you look, you will find a bar domain protein, more or less curved, but you will find a bar domain proteins. And an important event where the bar protein are found is during endocytosis. So I remind you, endocytosis is a process where a cell is able to incorporate in something. It could be protein, it could be lipids, and it can happen through the formation of this clathrin-coated vesicle. And if you see what the different steps of the vesicle formation, and you recognize here this very characteristic clathrin coat, you can see that when the vesicle is just forming, it's just, you see that you have protein with a shallow curvature, like this FCHO protein or FBP17. So at the beginning of the formation, you have protein with a shallow curvature. And if you continue and you are reach the point where the vesicle is formed and not detached yet, you have proteins with a more curvature which is higher. So it means that clearly, in this condition, at this type of concentration, there is a matching between the shape of the neck of the vesicle here and somehow the structure of the protein. And the question is how we understand that and how we can study that. So I remind you that, and we heard now this morning from Marcus a very beautiful demonstration that if we omit the tension term, the energy of bending per unit area for the membrane is given by this expression where we, in this case, C was G, but beside that, it's the same thing. So we know that the bending energy is reduced by this quantity here, which is the spontaneous curvature of the membrane. And I showed you yesterday, at least in a simulation, that it was proposed that the spontaneous curvature of the membrane depends linearly with the concentration of protein on the membrane. And we have a coefficient, which is a kind of a phenomenologic coefficient, which has a dimension of a curvature, which is called the spontaneous curvature of the protein. And this quantity, of course, reflects the structure of the protein. So the more bent the protein, you expect that the higher the curvature, but it also reflects, let's say, the local deformation of the membrane. So it's a parameter which will explain how much membrane is deformed when the protein is bound. So this is what at least was shown by Felix Campello and Misha Koslov for the amphipathic helix. And the question is, does it hold also for bar domain proteins? So, and what is also at least easy for you to realize that if now you have a membrane which is bent, imagine that you have a tube or a vesicle with a given curvature, C, you see that now an easy way to reduce the bending rigidity, the bending energy of the membrane is to increase the concentration of the protein locally. So then in this case, you will easily reduce the bending energy. And the more you concentrate, the less energy you pay. So you can easily feel that if you have this kind of coupling between the spontaneous curvature of the membrane and the protein concentration, you expect that as soon you will have a curved membrane, you should observe an enrichment of protein which have a non-zero spontaneous curvature. So how can we test that? So now I introduced yesterday the lipid nanotube, and I showed you with the lipid nanotube it's possible to control the curvature of the membrane simply by changing the membrane tension, and changing the membrane tension by, if we have a macropipette, by aspirating the vesicle. So you can imagine that if we have an experiment where we pull with our optical tweezer, you pull a nanotube, you have the vesicle held by the macropipette, and you inject proteins, like bar domain proteins. So you expect that this, if the proteins are fluorescently labeled, you expect to see an enrichment of the bar domain protein on the tube. And you expect even that this enrichment should increase with the curvature of the membrane. So and this you expect to at least to observe at low concentration, not in a regime where you get this tubulation. I'm talking in conditions which are more or less those of cells, so some concentration which are at high dilution in a sense. And what you can do if you have a confocal microscope, you can label your lipid with one color, it could be red. You can label your protein in green. And now what you can do is measuring the enrichment of the protein. Enrichment will be the density of protein in the tube as compared to the vesicle. And so this you can measure directly by measuring the fluorescence of a protein in the tube, normalized by the fluorescence of the protein in the vesicle. And to get rid of any geometrical aspect, you can normalize everything by the same quantity, but for the lipids. So if you measure that, you will have the enrichment of protein in the tube. And you expect that if this quantity is larger than 1, it means that you have enriched the protein in the tube. And as you understood this morning, that the curvature of a tube is 1 over the radius of the tube. So what we want to do is to measure this kind of quantity at steady state. Right. So another way to do, I'm not going to detail that, but has been done by Dimitri Stamou. You could also imagine that you can prepare vesicles with different size. You could tether them on a surface. You could have exactly the same thing, red lipid and green proteins, and you could measure the fluorescence of the vesicles in green, normalized by the fluorescence in red, and as a function of at least the fluorescence of the lipids, which is proportional to the diameter of the vesicle. And this is what was done for some bar domain by Dimitri Stamou. You can see that is already in this case that you see that the enrichment, in this case on the vesicles, is increasing when you reduce the diameter. But I mean, in our case, we think it's easier to do it with tube. Yes.
Both these experiments are getting at curvature sensing, right?
Yes.
But bar also does curvature like enhance.
Yes.
Cooperatively also does.
You can so they are coupled. You cannot decouple, they are coupled. So I'm not going to discuss too much, maybe toward the end, to the fact that at the time you get enrichment, of course, the protein tends to deform the tube to reach its optimal curvature. That's exactly the point. So in fact, there is a big confusion in the field, especially by the biologists, but they tend to distinguish protein which sense and protein to induce curvature. I think it's a two sides of the same coin. It's a question of concentration most of the time, okay? So in this case, I mean, this is an easy experiment to do, but it's not what I'm going to describe. So what we are going to see is an experiment where we have proteins binding to the vesicle, to the tube, and we are going to measure the enrichment as a function of the reduction of the tube radius. And you can imagine that in this case, of course, you will enrich the protein in the tube because you reduce the bending energy. Because you increase the spontaneous curvature of the membrane. But on other hand, if you locally enrich the protein in your tube, you will pay you have to pay a penalty, which is a mixing entropy, right? And the mixing entropy, since we have protein, I mean, is not too small, but not too big. So you can have an enrichment. If you do the same experiment with lipid, you will, I mean, I have no time to show you, but if we can discuss later, that this kind of sorting experiment is much less efficient because the lipids are very small. So the translational entropy is much more important. But in the case of a protein, at least you have to take into account the mixing entropy, but at least you will see that you can get some enrichment. And in the case of not bar domain, but protein which are embedded in the membrane, so I got this question yesterday. So you expect that if you have rigid inclusion, you might change the bending rigidity. So you have to take into account this stiffening of the membrane, and I will show you an example afterward. Okay, so that's the principle. What do we expect? So if you write down the bending energy, the mixing entropy, and you calculate eventually what is the total energy of the system, you can see that you have different terms. Here, what we have is a term which corresponds to the bending, and you recognize here the C square. You recognize the CP of the protein, the density of protein on the tube, and the tube radius. So that's a term, in fact, which comes from the mixing entropy, but eventually you can have it here. And you have a term which corresponds to the mixing entropy and, of course, the tension. And this corresponds to the force which is required to hold the tube. And this term chi is a kind of like an effective osmotic coefficient. What you have here, this is a secondary derivative of the mixing, the translational mixing entropy of the proteins, and a term here which correspond to the bending of the membrane. So basically, that's the term. I mean, I didn't want to go to the math, so I give you the expression, but you can find that in this paper. That's typically the term that you have if you consider a linear coupling between the concentration of proteins, the concentration of the proteins, the density of protein, and the curvature of the membrane. So you can from there minimize the bending energy. And what you get something which is kind of simple. You get an enrichment, which is 1 plus a constant divided by the tube radius, or at least the curvature. So you see that you expect to see a simple linear relationship between the enrichment and the curvature. And we have found 2 limits in the system. So in the case where we have, we have, let's say, a few % of proteins on our vesicles, we see that we have an expression here where we find, we have something which depends on the spontaneous curvature of the protein, but also on the density of protein on the vesicle, right? So we expect that the more protein you have on the vesicle, the lower the enrichment, because what we measure is a relative enrichment. Okay, but if now we go to condition where we have a very low density, and sometimes we have something which is almost at the limit of the detection, so we can still have an enrichment in this case, but you can see that the enrichment is independent of the density. We expect to have it independent of the density of protein on the vesicle, and you can see it depends also on the curvature, but you see that now we expect to get CP here. So we can test that experimentally with our bar domain, right? So, and then we have different possibilities, because I told you that the bar domain could have a positive curvature. So in our condition, following the Desserneau rule, the positive curvature is when the membrane is bent this way. And so if you have protein with bar domain that you inject outside of the membrane, and they will bind to the membrane, they will be, and you expect them to be enriched outside of the tube. And if you have an inverted protein, if you want to study this enrichment, you need to encapsulate into the vesicle. And now they will be experimenting a negative spontaneous curvature, right? So I will show you experiment first with a positive curvature. Oh, yeah. So what we have done is this protein amphiphysin, which has an N-bar domain. We have injected the proteins outside. The proteins, the vesicles contain negatively charged lipid. And what you can see in this is dead. Is it dead? I have another one, but then, yeah. You have another one? No? I don't know. Doesn't react anymore. I will take mine, but then I have to stay next to my computer. Okay, I will do that. I will stay next to the computer. This one is dying too. Oh, bon, that's complicated. That's complicated. So what you can see here, anyway, that you see that the vesicle is almost red, so meaning that we have a very low density of protein on the vesicle, but you can see that in its condition, the tube here, so the protein is green, and you can see that the tube is highly green. So it means that the protein is enriched into the tube. And you can do this systematically. So you can see that we can measure the enrichment, S, as a function of curvature. And you can see that for this density, which is like 1.4%, so we see that we have a very nice linear relationship between the sorting and the curvature. And from the slope, you can deduce the spontaneous curvature, the effective spontaneous curvature of the protein. And in this case, we find a very high curvature. We find almost 2 nanometer minus 1. So you can do that also in the limit where we have even less protein. So in the case of the vesicle I showed you before, so here we have different vesicles with different, a priori densities. And you can see that we have also a linear variation, and we find even a stronger curvature. And when we saw that, we were wondering we should probably compare that to what has been measured with the tubulation experiments. And at the time, we were a little bit perturbed because we found that the spontaneous curvature, which is found by the tubulation, is much lower. Almost 1 order of magnitude lower, right? And the radius was 11 nanometers. So in this case, our interpretation at the time was to kind of think that since this protein is not only a bar domain, but contains this amphipathic helix, maybe what we probe here is the insertion of the amphipathic helix in the membrane, which induces a very strong curvature, and not the bar domain themselves. So the value that around 1 or 2 nanometer minus 1 is what Felix Campello and Emisha Koslov predicted for amphipathic helix. So at least we saw that, I mean, practically the theory was fine. We have a linear variation, but the parameters that we find with that were a little bit perturbing, right? So that was what we were doing with a positively curved bar domain. And a little bit later, we started to try the same experiment with negatively curved bar domain. So this kind of protein, which is called ERSP53, which has an inverted bar domain. So we did the same experiment, but now we had to encapsulate a protein inside the GUV and to pull a tube. So we were also using a negatively charged lipid. And you can see very clearly that the proteins is very enriched in the tube. So it means that at least the principle works also for negatively curved membrane. And the question is now what happens if you plot for the same type of experiment, we change the curvature by aspirating the vesicle. What is the enrichment versus curvature? And then we find something which was very different. We find something which is, you can see here, this is a sorting, again, the curvature of the tube. And you can see that we have an increase with the curvature, but we can see that we have a kind of maximum, something we didn't see with the amphiphysin, right? And so it's not linear. Clearly, it's not linear. We have a maximum. And clearly, our model is not able to predict something like that. So we did that for different concentrations. So you see that at least we see that the relative enrichment decrease when we increase the concentration of protein in the vesicle. So you see that when we reach 5%, we still have an enrichment, but with a much lower and relative enrichment. But at least in all cases, we see that we have this kind of maximum in our curve. So it means that we had to design another model. So this is again something which was done by Andrew Cala-Jones, and what he did, he decided to have different terms in bending energy, and not to use directly the linear relation between the curvature and the density. What he did, he expressed the effect of the protein on the membrane by this term. So here what we have is the bending energy of a bare membrane, but you have this term here which corresponds to the interaction of the protein in the membrane. And he proposed that you can express that as a penalty, that when the curvature of the membrane is different from the spontaneous curvature of the protein, then you pay a penalty. And the cost of this penalty here is this parameter here, which is called kappa bar. So it has nothing to do with the Gaussian curvature, but that's a kind of energetic penalty to have a curvature which is different from the spontaneous curvature of the membrane. And you see that you have a quadratic term. So that's the way it was introduced in the model. And of course, you have a stretching term and a mixing entropy. Okay, so with that, we have a different parameter. We have the size of the protein, CP bar, and this parameter here, which is a phenomenological parameter, which is kappa bar. So, and we also suppose that there was no direct protein-protein interaction. So if you, again, minimize this energy, for a low density of protein, you see that in this case, you can tell it's no surprise. We put by hand that we should get a maximum, so we get a maximum. But so we expect to get a non-monotonous variation of the sorting versus curvature here. And we see that from this expression, we expect that the sorting should be maximum when the curvature will be equal to the spontaneous curvature of the proteins. So the question is, does it fit our data? And so that's what you can do, at least if you are working at low density. Otherwise, you need to solve this numerically. So question is, how does it work for the data that we have? And you can see here that we have the 3 sets of data with 3 different types of density, and all these data have been fitted with 2 parameters. One is the spontaneous curvature, of course, of the protein, and the other one is this phenomenological parameter kappa bar. And you can see that globally, the fits are quite good. And the parameter here that we get, and also something I should mention, that the maximum here is also correspond to this value, 18 nanometer. And in this case, now we can compare this parameter with the structure of the protein, or at least with what people have done with the tubulation. So I remind you, in this case, we expect to get tubulation toward the interior, and the radius which they found was 19 nanometers. So it means that at least the fit is good. It represents the physics that we have, and in addition, the parameter that we extract is okay. So it means it's a good model. Yes. I'm going to show. So we revisited our data after that, because we at least we had a kind of a global qualitative agreement, but we were not very satisfied by the spontaneous curvature that we deduced. So we just revisited our data. So it's possible sometimes to republish your data and with a new model. That's possible. So what we did, it's exactly what we did. We take the data from amphiphysin at low density. I showed you this data that we were fitting before with a linear variation. So we compare, in fact, the fit. The fit is better with this one. And if you use this fit, now you get a spontaneous curvature, which is 1/10 nanometer, which is much better, right? So, and I remind you that that's now more in agreement with what we expect at very high concentration. So, and in addition, so I don't have time to show you, but to answer your question, so we were doing experiments at very high concentration with a tube, and in this case, you can measure the tube diameter. So if you have a high concentration, what you find on the tube is a diameter which is more or less this one. So I think everything is more or less consistent, right? Okay, so we did that for different bar domain proteins. Amphiphysin, I showed you that. We did endophilin, which is another protein which also found at the last step on the endocytosis. We also find spontaneous curvature of the same order of magnitude, and this protein also 13-hexynian. You can see also, I show you a plot, and we find something which is not far too. So this, if I go back to the endocytosis, I remind you that all these proteins, amphiphysin, endophilin, 13-hexynian, are found during endocytosis at the late stage. And it turned out that the diameter of the neck of the vesicle before scission turns out to be of the order of 10 nanometers. So it means that in condition in cells that the protein are recruited, they are recruited, sequentially, because their curvature is more and more, it's higher and higher, matching the size of the neck during endocytosis. So at least we think that at least it's a signal that cells, or at least biology is using, or whatever is using physics, somehow to localize the right protein at the right place. Because I mean, this protein here, in addition, I have no time to show you, but they have a domain which is able to interact with a protein which is cutting the membrane, which is called dynamine. So they are just recruited at the right place at the right time, and they help to recruit the protein which will eventually cut that neck into kind of free the protein into the solution, right? So that's a kind of a physical signal.
Have people put like intrinsically curved proteins into biomolecular?
In what?
Into condensates, like
maybe they go by themselves. I don't know. Will? I don't know. But the curvature I'm talking here is small compared to this. I mean, the condensate that we see macroscopically, they are more like macron size. Yeah. So these are more like, we are talking about tenths of nanometer maximum, right? So, but yeah, so I don't know, in the condensate, people don't care too much because there is no membrane anyway. So where do you go?
People's partitioning to the surface, they control like the size.
Yeah, but there is no membrane in condensate. You have that in lipid droplets. Lipid droplets, there's droplets which are covered with one monolayer. So maybe on some of the lipid droplets, you have some protein which are recruited, but I think nobody described any bar domain. But on the condensate, that's not the place for the membrane proteins. Okay. So I should, yeah, I'm completely fine.
Atom and RP3 up there. Do you notice the feedback?
You mean this?
Is there some feedback? Well,
Which type of feedback do you think? So the fact, you mean, the protein help to deform the membrane? Would be your question?
like, for example, where the vesicle buds off. I
Yes.
guess you cannot have that filament directly.
You mean this actin here? Yeah, so I think that the role of actin at the time of the point of the fission is still not clear. I mean, it's especially in human, at least in mammalian cells, it's not so clear. So I could not comment about the actin in this case. More question? Yes.
So the largest diameter used in data is about 100 nanometers. So is there a lower limit as well for the sensing from the flat membrane? Like, is there a lower limit after which the sorting happens?
That's a good question. In fact, in our case, we consider that we have a significant sorting when we reach a factor at least 1.5, at least. But there are people who did experiment with cells, that they put cells onto nanofabricated structures. It could be pillar, it could be rods with a curved area. And in this case, the best they can do is 100 nanometer. And few hundred nanometer, and they have seen that if you put cells and you label the some bar domain proteins, you can see that the bar domain protein are, let's say, a little bit enriched, because what they find is a 20–30% enrichment, so it's not much. So it would be like 1.3 for me, but at least they have seen that even so, it will be really the tail of the curve, right? And even though in this condition they have seen some enrichment. More? Okay, so that was for the bar domain protein. So now we can go to inclusion. So the question is, can we see the same type of effect with protein which are embedded into the membrane and which have a conical shape? So I showed you yesterday, when I was showing that protein can deform membrane, I showed you that you could have some transmembrane protein. So in this case is a protein with a V shape here, which is embedded, as you can see, in the bilayer. And the protein have a conical shape. And you can see that if you have reconstitute them in small liposome, which should be spherical if there would be no protein, you can see that the liposomes become tubular, right? So, so it means at least the membrane protein can deform membrane, for sure. And so that's a Cosgrain simulation from Peter Tillman, where he was studying the deformation of the membrane by many membrane proteins. So you can see here that they can have a different type of shape. They could be very big. They could be, very big cytosolic part here. The protein I'm describing is here. And you can see that here he represented the local membrane curvature. So that's a very small patch of membrane. So he has a 4 protein in his patch. And he described the local deformation by this quantity here, which is the mean curvature of the membrane, right? And you can see that you depending on the protein, you can get more or less blue or red. So blue is negative, red is positive. You can see that it's more or less isotropic or anisotropic, depending on the protein. So he described that at least as a fact that each membrane protein has a kind of a fingerprint and has a singular deformation of the membrane, right? So we were not using all of them. We were trying to compare 2 proteins and to see if the protein could be enriched in a curved membrane. So for that, we were using 2 proteins. So as I say, one is a voltage-gated channel. It's called KVIP, and it has a conical shape. And we wanted to compare that with a protein which does not deform the membrane, which would be something like this one, not much. And this protein is aquaporin 0. And of course, Peter did also simulation for these 2 proteins, and you can see that this protein aquaporin 0, almost no deformation, cylindrical, and you have this protein here, which has in average a conical shape, and you can see that maybe it wants to reply to Marcus yesterday, at least maybe the proteins have no shape, but at least they have an imprint in the membrane and they deform the membrane. And you can see in this case that this protein is deforming the membrane. So if they form the membrane, it means there's a non-zero spontaneous curvature, right? So the question, are we able to measure that? So of course, we had to do experiments where we reconstitute the protein in a giant vesicle. So it's more complicated than having the protein in solution. You have to work a little bit harder, and you do the same thing. You put a tube, and you look at the enrichment. So in this case, it's a little bit more complicated than the case of the soluble proteins. Because when we reconstitute the proteins in the membrane, especially if they have a conical shape, the problem is they are not oriented in the same direction. The way we prepare them ends up that we have protein which some of them are facing inside and some of them are facing outside. So they are kind of symmetrically oriented. So it means that if they have a spontaneous curvature altogether, and we are 50-50 of the orientation, altogether the total spontaneous curvature will be zero. So it means in our model, if you take in this case, if we take the linear coupling between curvature and density, at least we need to put a higher order because the first order will be zero. Okay, so you have to include that. And in addition, you can imagine that if the protein now is in the membrane, the bending rigidity of the membrane might be changed, especially if you enrich the protein into the membrane. So, there was a model which was developed by Andrew Caladron and Gil Toombs, and basically they make a very simple assumption. They propose that the effective the bending rigidity of the membrane is simply the addition of the bending rigidity of the bare membrane and of the something which would be an effective stiffness of the protein, which they call kappa protein, and corrected by the fraction of the protein that you have in the membrane, right? And of course, you have to take into account the mixing entropy like before, right? So if you put everything in the equation, so now what we have is they were using the simple, at least the linear relationship between the curvature and the density with an higher term, and this bending rigidity, which depend on the density. So if you do that and you minimize the energy, you can calculate the entropy, the enrichment, and you can see we have a complicated expression. So you see that we expect the enrichment will depend on the square, again, of the curvature, right? You see that it's not linear in this case because of these two aspects, the fact that we have this symmetrical insertion, and the fact that we have also this stiffening of the membrane by the protein. And you can see that we have different terms. So here, so we have of course, the density of protein, which appear here. We have the size of the protein, at least, which is the molecular area of the protein, which is AP. We have the bending rigidity of the pure membrane, which is kappa L, and the effective bending rigidity of the protein, which is kappa P. So that's a complicated expression at low concentration, at low curvature, but still you can feed the data. So what type of data we have? So I will show you first the experiments. So you have, again, the protein which is conical, the protein which is cylindrical. So you can see that here we have in green the KVAP and lipid in red again. And you can see that when we have a yellow signal, mean that the fluorescence of the green and the red are equivalent. But you can see that in this condition, the tube is green, meaning that we enrich KVAP in the tube. So even though we have the two insertion, eventually when we pull the tube on, I mean, clearly we have an enrichment, which was not given for granted. And you can see that if you change, so you change the aspiration, so you reduce the tube radius, you can see that the relative signal of the green is increasing. Now, if you compare with what happened with the cylindrical protein, you can see that the tube is yellow. And you can see that when you reduce the tube radius, you also reduce of the same quantity the signal of the protein. So it means that at least that's correct. So if you have a cylindrical protein, which normally does not deform the membrane, it's not enriched in the tube. And you can see that even though the protein are oriented both ways, we can have a sorting of the proteins. So now what do we get in terms of result? So we can calculate the sorting as a function of curvature. And we can see here that we have two to density, you can see that when we again, as for the high bar domain protein, we see that when we decrease the concentration of protein, we increase the concentration of protein in the vesicle, the relative enrichment is decreased. And we see that we have a non-monotonous variation of the sorting. It looks like what we had with the high bar or with the bar domain, but I think the model is a little bit different. And we can, of course, calculate the spontaneous curvature of the protein. This is one of parameters of the fit, and we find 1/25 nanometer, which correspond to, I mean, if you try to make a very rough geometrical estimation, it corresponds to a conicity of 5 degrees. And also we can, from that, measure the stiffness of this protein, and we were very surprised because we found a stiffness which is only 50% higher than the stiffness of the membrane, which is what we found. And okay, so it means that at least that's fine. So the question is, is this number making sense or not? So I think again there was some simulation, MD and coarse grain simulation done by Rainer Buckmann, and he was trying to simulate the same protein that we were studying. So he didn't take a big piece of membrane, he took a bicell, so it's like a piece of membrane but limited, and he was incorporating one protein in his bicell, and he looked at the deformation of the membrane induced by the protein. And you can see that if you have the conical protein aquaporin 0, it's effectively not deforming the membrane, but you can see that if now you have the KVIP, you see that the membrane is bent, and you can calculate the spontaneous curvature of that membrane, the equivalent of this piece of membrane, and you find something which is very similar to what we have found with our tube pulling experiment. Yes.
High density, the sorting is reduced.
I mean, it means that if you look, the sorting is a ratio between the density of protein that you have in your tube divided by the density of protein that you have on your vesicle. But so it doesn't mean that the density of protein on the tube is lower. It means that the relative enrichment is lower, but the density is high. But since you divide by the density of protein in your JV, you end up with a relative enrichment which is lower. No, that's simply because the more protein you have, the more you bend the membrane. That's only, and also no, that's why you enrich. Why you get this maximum is because if you get too much, you pay now since the protein is a little bit stiffer than the membrane, then you start to pay in terms of bending rigidity. So of course you get an enrichment because of depending also on the density. But there is a maximum which is also coming from the stiffening of the membrane.
Be a limit to how much we can get into the tube as well, right? That would reduce the efficiency.
Yes, yeah, that's true also. Yeah, and there is a point also where you have also to take into account the interactions. So I think that there is a limit to it. So I think that this kind of model is working when you have a few percent of protein. 10% here is already a little bit extreme. Huh? Fine? Okay, so much faster than I thought. Okay, so the message of, at least what I tried to convey so far about curvature and protein with certain spontaneous curvature is that at least the membrane curvature can induce a protein sorting. And this is related to the protein effect, what I call effective shape. I mean, the effective shape meaning that it's included the effect on the membrane. And I showed you that we have I mean, at the end of the day, we can have two types of model. I mean, from the simulation, from the calculation from Misha Kozlov, or from our experiment with transmembrane protein, we see that at least this linear coupling is working relatively well if we have insertion. But in the case we have protein with a curved shape and they are bound to the membrane, I showed you that we need to take a model with a it is what we call the curvature matching model, which works better for this protein. And interestingly, I think I showed you that we can, from very macroscopic experiments, because tube pulling is a macroscopic experiment. We use a confocal macroscope, even an optical tweezers. We can directly from there deduce information which are molecular. We get an effective shape, which we can relate to the structure of the proteins. And I think also what is very interesting is we get that in a lipid environment, because in many cases, when people do a structure, they do a cryo-EM, or for that, what they have is usually they have the protein in detergent. So I think that we can get this information in a much more natural environment for the proteins. Okay. Okay, so now I'm completely switching topics. So if you have questions on that, well, better to continue.
Regarding the mechanism of sorting, we know it happens because, say, for example, we continuous curvatures. Is it flowing along the membrane, the region of high curvature, or is it binding?
That's a good question. So I think it's probably both. So I don't know if it's a question of K-on. So the question is, it's a question of K-on, which will depend on the curvature. Or is it a question of redistribution of proteins? So in our experiment, in our model, we don't have the K-on dependent on the curvature. We suppose the proteins are bound and they redistribute, so they diffuse. They are free to diffuse, and they diffuse from the vesicle to the tube. In the case where you have small liposomes, that in this case is more you don't have the same system. And it turned out that we have a very big discrepancy between the data which have been obtained on liposomes and the data which have been obtained on tubes. And it's even more subtle than that. For the bar domain protein with an amphipatic helix, since I have time, I'm explaining. So it turned out that when you have an amphipatic helix and you have a tube, there are some EPR experiments showing that in this case, if you have the tube, your protein is really touching very tightly the membrane and inserting the amphipatic helix deeply. So it means the effects of the amphipatic helix is small. If you have a sphere and the radius of the sphere doesn't match well, the fact that you have a sphere, in this case, the protein interacts through the amphipatic helix. And that was also shown with EPR. In this case, what you feel, even though you have a bar domain, the only thing you feel is at least the effect of the amphipatic helix in the membrane, what I described yesterday. So, and this discrepancy, at least for a while, we were worrying because we didn't find at all the same results. And I think that's a reason. So I think in this case, in addition, when you have the domain, the proteins in solution then bind, so you reach a steady state, but you probe more the K-on and K-off if there is a K-off, right? Yeah. Good? Okay, so I'm completely changing topic. So I showed you, I was addressing the point where the protein is binding to a different neck, size of the neck, depending on curvature. So it's what I tried to explain for, at least for this part of my talk. The question is now, how you cut. So if you have, I put it here, if you have a vesicle like that, or you have a tube, the question is, how do you cut? And it's a fluid membrane. How do you think that you can cut that? If you start to pull on this vesicle, you know what is going on. You are going to pull a tube, right? Because it's not on pulling. Okay. You pull on the tube, you are going to extend the tube. So you cannot, in this direction, you cannot cut the membrane. The only way to cut a membrane is by, you can imagine, is by constricting the neck. If you point that, but let's say the two cytosolic leaflet of the membrane will touch each other. You can imagine in this case, you might have a fusion between these two monolayers, and you can get a fission. That's one way. So by constriction, you can imagine you can cut it. So you can constrict that. You can constrict that. Maybe you can get a fission. There is other, but it's not the only solution. But it's not by pulling; it's by constricting. And there are other ways to pull to cut a vesicle. So I think I kind of summarize different aspects here. So to constrict, there are different ways. There are some enzymes which are able to form polymers. So this is a case of the clatrine vesicle. there is a polymer which is called dynamine, which is recruited at the neck, helped by the bar domain protein, recruited at the neck. And when you use GTP, it changes its diameter and is able to constrict the neck and to cut. There is, let's say, even more physical way to cut membrane is to use line tension. So if you have two domains you have a different type of domain, so different type of composition in a membrane, you can have at the boundary of the domain, you can have a line tension, and this can induce a constriction and can also induce fission. So this is again a constriction. There is an even more subtle way is that if you have this kind of structure here, which has a Gaussian curvature, non-zero Gaussian curvature. Now you know what it is. If now you insert like the amphipathic helix, amphipathic helix tends to create locally a positive curvature. And I mean, I have no time probably to show you that, but in this, it has been shown that it's able to destabilize the neck and to cut it. And I'm going to show you something a little bit different based on this bar domain protein, which in fact use the fact that if you increase a lot the tension in the membrane, you are going to create a pore. I mean, I showed you yesterday, if you increase the membrane tension up to a point that you reach 5% increase of area, you create a lysis of the membrane, you get a pore, and this is enough to break membrane. Okay, so I'm going to talk about that actually. Okay, so you have seen that, I've described endophilin. It was an endophilin in the context of clathrin-dependent endocytosis, but it turned out that endophilin has also been shown to be involved in clathrin-independent endocytosis. And you can see here that's a movie where you have a toxin which is endocytosed through clathrin-independent. And I don't know if you can see, but it doesn't matter. The movie was to show that at you have some tubules which are corresponding to the endocytosis of this toxin, which is in red. Maybe this movie is not very... it's starting again. Yeah, maybe I can. So basically, if you follow let's see if I manage. Yeah. You can see that you have tiny, too much light, but anyway, you have tiny tubes which are in red, and you can see that the endophilin, which is in green, is exactly localized at the position of this tube, right? And so in addition, there was evidence that in the case of toxin or in the context of some receptor, you find tubules which are formed for the endocytosis, and you find endophilin on these tubes. It turned out that now in biology, what you can do is can delete the protein and see what you get. And what you can see here, that what happened in this kind of cell, if you have endophilin, you can see that you have tiny, you can see in this dot point correspond to the toxin labeling. And you can see that you have some endocytic bud, but they are very tiny. And you see that if you remove endophilin, the tubes are longer. So if the tubes are longer, meaning they are not cut. That's the idea, right? And so that's one of the evidence that endophilin could be involved in scission. So after what we saw, that was a little bit surprising. So especially it was surprising that now I'm going to show you an experiment. So compared to the experiment I was doing before, we were working in condition where we have let's say a few % of protein on the membrane. In this case, we are ending in conditions, so we inject a much higher concentration in the bulk, and we end up with a higher concentration in the tube, right? And if you do that, so you have originally you have a very big tube. You can see the diameter is huge, it's 100 nanometer, 120, and we are going to inject the proteins with a pipette, and you see what is going on. So you see that the protein is start to assemble. You can see that the tube radius becomes almost not detectable. You see that you get a decrease of the tube radius. You go from 100 down to 10 nanometer, right? So I will play this again, hopefully. So you see that when the protein is binding, you have a high signal, and you see that the tube is almost not detectable because the radius is very small. So it means, and you see that the force at the same time decreases, and it's not because the tube is broken, it's because the tube is stable. And so the force at the end of the day is 0. So it means that the membrane, the proteins makes a scaffold, which now requires no force to hold the tube. And if you look at the radius, you find something which is exactly what we found with the sorting experiments that I showed you before. So that was surprising because it showed that the protein is not breaking the tube, it's stabilizing the tube. And it's also in, I wanted to show you this simulation. So they were done by Mayo Simonovic. He was a PhD student with me doing experiment and with Greg Voss doing simulation. And he was studying the assembling, that is the organization of this protein where they were on a tube. So he formed a tube, he put the protein on the tube and he looked how they organized them. And you can see that they spontaneously organize. They don't interact together. They just self-organize into a very nice helical structure, right? So again, this is the kind of structure we stabilize the tube in our experiments. And so he showed that depending on how much protein you have, I mean, you can have at least they are very kind of loose. I mean, at low density, but as soon you reach a density which is high enough, you get this kind of scaffold. So that's at least not showing any fission. And more or less, I mean, this was very, very well known in the literature that if you incubate your liposome with protein, what you get is tubule, and these tubules are stable. Okay, so the question is, how can we understand that? So turned out that, yeah, that was the big question for us. So it turned out that if you discuss with our colleagues biology, they realized that this tiny tube, which are formed by the toxin originally or by the receptor, usually they are very tiny, but they need a protein which are called molecular motors. So that means that we have a kind of a tiny structure which is formed by toxin, for instance, but you have molecular motors which are walking along macrotubules, and these molecular motors are involved in the extension of this tube. If you don't have any molecular motor, you don't get any endocytosis. So clearly that we need something which not only deform the membrane, but we need something which is even extending the membrane in the presence of endophilin, right? This is exactly the cartoon we had in mind. So at least what our colleagues had in mind. So you have a tiny bird which is forming, probably coated with endophilin, and you have some molecular motor which bind and extend it. And at some point, there is a fission. So it means that in a static way, the scaffold is stable. The question is, now what happens if we dynamically extend it? And we can do it because we have our optical tweezers, so we can do that. So we can pull on our endophilin-coated tube. That's easy. So what we can do, so now we have the experiments where we have already coated our tube with endophilin, and now we can pull on it. We can replace the motors by moving the vesicle away. So you can see, hopefully here, that we pull, the force increases in this case, and there will be a point where the tube is breaking. And so you can do that at different velocity, and we find out that you don't need to be very, very fast to break. What you need is to go over 20 to 30 nanometers per second. And typically, the molecular motors, when they walk along macrotubules, they easily go to 100 nanometers per second. So it means that at least experimentally, we can reproduce by pulling, we can reproduce the fission of the tube in vivo. So the question is how it works. So if you do the experiments dynamically and you have no protein on your membrane, you can see here that this is the force versus time when you extend your tube at different tension, at different velocity. You can see this is a very high velocity, is 1.5 micron per second, but you can see that the force remains constant. And I told you yesterday, you can extend the tube as long as you don't go very, very fast. You don't change the force over time. So you extend or versus length. So it's completely stable. Okay. So it means that the scaffold does something. The question is what? And what you have to keep in mind that when you extend the tube, so you have your vesicle, it's like a flat membrane, and when you extend your tube, you have a flow of lipid, of course, which is advected by the extension of the tube, which eventually hit the tube, right? So, so this flow of lipid is very important. So it means that the flow of lipid has to be as fast as the velocity of extension, right? The question is, if you have something which is perturbing the flow, and I wanted to show you that. So I show you a movie where we have the full coating of the proteins on the tube, but we have cases where we don't have a full coating. Sometimes we have only a partial coating, like here. So this is a chemogram. So basically what we have here, every line here corresponds to one snapshot of the tube for the protein channel and for the lipid channel. And you can see that here we have we don't extend the tube, so we have the endophil here. We see that we have a very strong constriction of the tube because the lipid signal is very small, okay? And now we start to pull, and we extend the tube here up to the point that here is broken. But you can see that here, this is a limit with the vesicle, and we can see that basically we have, let's say, we remain, the scaffold of protein here remains connecting to the GUV here, until it breaks. So just to tell that sometimes we have a partial scaffold. So the question is, what is this scaffold doing to the lipid dynamics, right? And one way to look at the lipid dynamics is to do what is called FRAP, so fluorescence recovery after photobleaching. So what you can do is you have your fluorescent lipid here, and you can illuminate so much that the fluorescence is dead. I mean, you kill the fluorescence of your lipid there. And if you have, by diffusion in this case, you have a recovery of the fluorescence, so it means that the lipids are free to diffuse. And if you do that without proteins, so this would be this graph here, you see that you bleach your tube here, and you can see that over time, after a few seconds, you get a recovery of the fluorescence because the tube naturally diffuses between the GUV and the tube. Now, if you do the same experiments when you have the scaffold of proteins, you can see that basically after 40 seconds, we have nothing. So it means that also already for diffusion, the scaffold perturbs the diffusion of the lipids, right? And you can also, now what we can do is you have the tube, you have your scaffold here, and you can change the tension. You don't extend the tube, but you can change the tension. If you change the tension, now if you have no protein, your tension will be equilibrated very rapidly because the tube, thermodynamically, the tube radius will equilibrate to reach the right diameter compared to fixed by the tension. But if now you have something which perturbs the diffusion, you will see that the tension is not equilibrated. And what we can see that what we have done here at time 0, we have changed the tension. So if we have a control, we can see that we have a rapid kind of equilibration of the intensity of the tube radius if there is no scaffold. But if you have a scaffold like here, you can see that if you change the tension, now if you look at the fluorescence of the lipid here, basically it doesn't change. So it means that after 4 or 5 minutes. So it means that clearly the diffusion of the lipid is perturbed by the scaffold. Yes?
The radius of your tube, is it constant everywhere, or does it change? Like where the scaffold is, it will be like the most positive curvature that it can go to, and where there is no scaffold, it will be back to some other.
It's like in our experiments, it's like even if you don't have a full scaffold, it's like the tube, the bare tube is kind of isolated from the vesicle. So at least the time scale for diffusion will be very long. So I think that you can do what you want to change the tension. The tube radius here doesn't change. Okay? And if there's a scaffold everywhere, the tube, it doesn't change as well because you have a scaffold. Okay, so the model we had in mind that now if we are going to pull when we have a vesicle, the scaffold here, and when we move the vesicle away. So we have the scaffold, which is limiting now the advection of the lipid into the tube to help to extend the tube, right? So to describe that, so again, there was a model by Andrew Cala-Jones and Jacques Pro. So we describe this effect as a friction between the scaffold formed by the endophyllin and the tube, right? So it means that, and of course, if there is a friction that we call psi, so it means that now when we are going to extend the tube with a velocity v, so we have a friction coefficient, which is effectively associated to the velocity of the pulling. And you can see from there that if we are going to pull fast, the tension should be expected to increase a lot. If we go slowly, we should have a less increase, right? And to test that, you remember that the force, I should just give you again the expression. So the force to pull the tube normally depends on sigma, at least for bare membrane, right? So it means that if we measure the force versus time, we should have something which reflects the variation of sigma. So if you do that, you can measure the force. So you measure the force before you pull. So you measure difference of force when you pull corrected by this initial force as a function of time at different velocity. And you can clearly see that you have two regimes. So basically, you have a regime where the force increases a lot. And we describe this as an elastic regime. So basically, when you start to pull, what you do is it's like you have a constant number of lipid and you extend this tube. So you have an elastic, like an elastic behavior of the membrane. And if you leave more time, now you start to force the lipid to go through the scaffold. So with a high friction still, but they are forced to flow there. And then you reach a regime where you have viscous variation, right? So if you kind of build up a model based on this observation, so this is the feed that we have here, right? And if you feed the data, you can extract friction coefficient. So the number doesn't matter much. We find something which is 80 Pascal second, but doesn't matter. But the good thing also with biology is that you can play with the protein. So you can play with the friction coefficient. So what you can do is manipulate the proteins. What we can do is we suspect that the endophyllin has the N-bar domain, has amphipathic helix, and the amphipathic helix are inserted in the membrane. So we suspected that the amphipathic helix could play a role in the viscosity, at least in the friction. So we can cut them. So we have a mutant here, the blue mutants where we have cut the amphipathic helix. And we can also make mutation to increase the interaction of the bar domain with the membrane. This is this mutant here. And what you can see that if you have you remove the amphipathic helix, you see that the friction decreases based on the same analysis here. And you see that if now you make it more attached to the membrane, you increase the friction and you see that you have a higher friction. Yeah. So the question is okay, we understand how the force increases, but how is breaking? And our suspicion is that it's breaking because of formation of a hole. So, and we saw that in our experiments, most of the time, the breaking took place if we have an incomplete scaffold, it was always at the edge between the scaffold and their tube. So we suspected that the scaffold was a place for the nucleation of the hole, right? And you can, again, derive a model, which is kind of a model where you describe the nucleation of the pore at the edge. So what we did, we suppose that to form the hole, there is an energy barrier. So if you have a hole into a bare membrane, you have the cost of the hole will be proportional to the radius of the hole and something which is the edge tension. But if now you form a hole because your tension becomes so high, in this case, you reduce the cost forming a hole, because you, in this case, you reduce the membrane tension by opening a hole, right? So basically, if you take this energy barrier, which takes into account the variation of the tension versus time, you have an energy barrier which depends on the time, and you can use the Kramer theory to calculate the probability to nucleate the pore with this energy barrier here. And if you do that, you can predict two things. You can predict the variation of the force, the force for the breaking as a function of velocity of pulling. And you can see that we expect a relationship with a scaling factor of 1/3. We see here the friction coefficient, the bending rigidity of the membrane, and also this parameter, which is this tube, this whole radius here. And we saw that we can also predict the time at which the protein, the tube will break as a function of velocity. So I will show you only that, but we tested that as well. So we checked if the force at which we observe breaking scale as 1/velocity 1/3. So you can see that we did that for the wild type protein. For the two mutants, the one where we cut the amphipathic helix and the other, and the mutant here, you can see that we scale, the scaling is perfect. We have a 1/3 scaling. And from that, we can deduce so this is the friction that we measure here before from the force increase. We have also the same tendency for the fission. So you see that now that we obtain a friction coefficient of 30 for the wild type, we see that again, we see that the friction from this kind of measurement, we see that we have a strong reduction when we cut the amphipathic helix, and when we have a, yeah. Yeah, it's bizarre. I agree. Yeah, I mean that we have, you know, we have 4 points. That's why. I mean, it doesn't mean anything. I mean, it's low. I mean, it's low. I agree. It means that. I agree. That's bizarre. It's true. I didn't. It's good. I never realized. I never saw it. That's interesting. Yeah, but I agree. So, but anyway, the point is not the absolute value, but the point is to, again, so you can see from the slope already, you can see immediately here that we get the same type of behavior for the mutant and the wild type. But again, you see that is consistent with the same type of theory, that we get this friction between the membrane and the tube. So basically, with that, I mean, I think it was at least an interesting type of fission. And then I think that now there are more and more, examples in biology where this kind of fission can exist. And to summarize the different steps which happen for the fission here, what we have is we have a coat. So the coat is made by bar domain, but it could be maybe made by other type of proteins. But we need a coat that changes let's say the advection of proteins of lipids when you extend a tube, or at least some transport intermediate. And so when you start to extend, so it's a dynamical process. So if you start to extend and you are above a certain threshold for the velocity, you have an increase of the tension, and the tension is increasing up to the point that you nucleate a hole. The membrane is not stable anymore, and then you get a fission. And I mean, usually I like to finish the story by at least a movie, which basically explains the physics of this system. So imagine that you have a spaghetti which is overcooked, and you put it in water. You can do that, nothing will happen. But if you put your spaghetti in a viscous medium, like honey, then you can have that. And then it's breaking. So it's exactly what happens, at least very roughly speaking, with our tubes. We have a high friction, which is represented by honey. And then basically this friction increases the tension, and the tension is high enough to induce breaking.
Breaks at the interface.
Most of the time. Yeah, but here there is no coat. I mean, so white coat, I don't know what happened in spaghetti where to decide where it's going to break. So I don't know. I was thinking I will manage just on time. So I wonder what I should do. I can continue on, or maybe you have questions. I will take questions.
discussion.
Yeah,
exactly.
yeah. I think it's better to discuss than, because if I start to discuss another type of a session, it will be too late. So, yes. I have to come because
With the first images we had with the tube formation with a high concentration of bars, do they have interactions with each other, or do they just happen to align in the right shape? Because
yeah, so the question is why don't they they bind homogeneously on the tube? No.
it has to be all aligned to get to be in a nice tube shape.
You could see in the simulation, at least, I don't know, but in the simulation, you can spontaneously self-organize. So the protein, they have this kind of anisotropic shape, and the best way for them is to be oriented perpendicular to the axis of the tube. So that spontaneously orient them. And so I think that's why they eventually they form an organized scaffold in a sense. But even though there is no direct interaction between maybe something I have time to show, because to explain this self-organization, maybe I could find the slides I didn't show on this bar domain, but maybe I have time to show them now. Yes, this kind of thing. Yeah, maybe this is simulation on vesicle in this case, right? So this is simulation where you have a vesicle and you have bar domain which spontaneously bind and self-organize. And again, it was Mio who showed that because this protein locally deform the membrane, and at least they tend to self-organize spontaneously, and they form a nice network, as you can see here. And this is what you get even on the flat membrane at low tension. And to show that that's directly membrane-mediated interaction, this tip interaction, you can see that if you increase the tension, this network disappears, right? And I think there was also simulation where people have looked at what happens if you increase the concentration. And you can see that depending if you increase the concentration rapidly or not. So if you increase the concentration rapidly, you can have even this kind of structures which appear. And you can even, I think there's something from Noguchi where he got, yeah, you can see what happens in this simulation. So you see the proteins spontaneously organize themselves in a kind of a stack, right? And then just eventually form tubes. This one? Oh, yeah. We don't see that. I mean, that's the problem is we don't have the resolution. I mean, that's simulation. So I mean, maybe we have many simulators here, but I think that the beauty of the simulation for me, if I trust the simulators, is that you can see the organization of the proteins that I cannot do on my vesicles, right? So in a sense, it's complementary. So in the tube, I mean, probably, and there are also some cryo-EM experiments which have been done at high concentration, so very high concentration, and you really see this kind of organization of the proteins. But we have shown that depending on the bending rigidity of the membrane, so if you have membranes which are stiffer, you have bigger tubes. So the protein, instead of being perpendicular to the axis, can be parallel to the axis. Okay. So, but at least they self-organized, one way or the other. Yes.
Here, the fact that this continuous curvature, the approximate speed linearly dependent on tension. And so my question would be, so as long as the particles are just coating the membrane, I see why. But once they start, you know what I mean, like forming layers of it, then I don't see why it should be there anymore. So
What do you mean layer? There is only one layer of proteins anyway, so they don't pile up, huh? No, they don't pile up. They conicote, yeah. There is a limitation. I mean, anyway. I mean, on the tube, there is a time where if you get a full, and we never reach a full coverage. We estimated that. So in the case of the epophylline, this is probably the most, at least the denser we can reach with this kind of experiment. We reach 50% of the surface. It's a lot, but still not as much. So and that's the maximum. And on the vesicle, if you get too much, then you start to get tubes, and then okay. But I think the protein never pile up, never. Now it's always, I mean, the density is always less than 1. The surface fraction is less than 1. Yes, Marcus.
Outside.
Yes. Yeah.
Just wondering now, that the lipids have to flow, but only the lipids on the outside are impeded and not the ones on the inside. We put the outside leaflet under a higher tension than the inside. Now that tension difference between the bending point
I showed.
inside. I wonder what that also helps.
Yes, but when we do the bleaching experiments, so if it would be only 1 leaflet, we could see a partial recovery. It's not what we see. So it might seriously depend on the composition of the membrane. So meaning here we have a very, I mean, we don't have a simple DOPC, etc. So we have a kind of a more complex composition. And it's possible that these two leaflets, even though one is immobilized by the coat, but maybe that we have a lot of interdigitation of the lipid. I don't know. I think that they are maybe strongly coupled, to put this way. At least we don't see any recovery of the lipids fluorescence in the, but maybe what you say is right, meaning that if we take a simple composition where we get maybe less interdigitation or less coupling, maybe we can be in a situation where we immobilize only one leaflet and maybe the other one will be free to diffuse, and that will be creating probably as different stresses. But it's not what we did. Huh? Yeah. Yeah, but so that agree, but it's true that to do this experiment, when you measure now the let's say the interbilayer leaflet requires usually to go to very high velocity. So we didn't do this kind of experiment. You're right. In principle, we should measure that an intrinsic, let's say, interleaflet friction of the bare membrane in the absence of protein, but we didn't measure that. We didn't do it. But in principle, that's fair. Yeah, we should do it. Yes.
Here's one decision. So you said you expect a pore to form.
Yes.
In this case, kind of violent case of and do you think that that could cause leakage? Is it how we expect it to work in cell?
Yeah.
And also these other mechanisms that you didn't have time to go through.
It's transcription.
Do we know if there is a pore?
There is a lot of debate about this pore. At least in our case, we try to see if, I mean, we try to evaluate if we, in this case, it could be a simple imiffusion process, etc. In this case, we really think that's a pore, and it's possible that you get, I mean, the pore life is probably very, very short. So you might have a leakage, but maybe it's very, very small. Anyway, we tried to see if there was a way to test that experimentally, but we didn't find a way to check short leakage. So that's at least we could not fail. But in the case of the constriction, so I remind you this dynamine story where you have a filament which is polymerizing, which is constricting. Some people claim that there is no leakage, and some people like what is his name in Spain? At least some people claim there is some leakage, a small leakage. You can have a small leakage, but I think that's still, for many good reasons, you can imagine that in cells, you prefer to get fission without leakage because there is a big problem, which is normally calcium. And if you get calcium going in and out, that can create problems. So it's not a reason, but at least that would be a possibility. But it's very difficult to get that. There is only one way sometimes to check that is with electrophysiology, because it's very sensitive to a very short pulse of, let's say, ions, if you have salts outside. So maybe I will remember his name, Marcus Vadim, yeah, Vadim Frolov, yeah. So I think he has evidence that you can have fission with leak and without leak, depending on the type of fission that you get. Yes.
Minimal threshold surface fraction coverage necessary that.
Depends on the proteins. So that's it. What we need is to form a scaffold. But we managed to get, we managed to get also fission with proteins which apparently do not form scaffold, but which are concentrated enough on our tubes. So that's the only thing you need is that, form a scaffold. So it means that it will be a case by case. Yeah. And it could be a scaffold, which you can have a scaffold. So we try to do the experiments for dynamine, for instance, without GTP, so not constructed, and you can pull, and you don't get fission. So it really depends how the scaffold, so you need a scaffold, meaning something which is dense enough, but it needs to interact strongly with the membrane to somehow perturb the lipid advection. Yeah. Yeah. US first.
Depends on the concentration. So,
I will repeat. She wonders about the spontaneous curvature depending on concentration.
yeah, and I mean, it can be positive or negative. I mean,
Positive or negative, yeah. So in our case, we found that it's positive. So what we define as a positive when it's on a convex, yeah. So this protein which bind to the membrane through, they have a positively charged here, and when they bind to the membrane, they bind to the exterior of the tube, because the exterior of the tube has, by this de Zerno rule, has a positive curvature, right? And if you, I didn't show you, but if you take the protein which has inverted curvature and you put it outside, and it can bind still to the vesicle. It's not that the vesicle is flat, but now when you pull a tube, you don't see any proteins with this negative curvature in the tube, right? But if you put it inside, now inside, for the protein, the curvature is right. So from outside is like a negative curvature, and now you have the protein is binding through its again positively charged residue, and the tube curvature has a right sign for that kind of proteins. Yes.
That's why the
we think that I think that is like if you think about defects where nucleation, for instance, nucleation of a crystal somewhere, when you get a defect, it's usually where you get the nucleation. So we think that the most probable place makes sense if it's at the edge, because anyway, if you are at the edge, you can have a hole forming, which is only half of the hole, right? So instead of the middle of the membrane when you make a full hole. So the probability, it's not always the case, but the high probability is usually at the edge of a coat, because it's just pay half energy. Yes, yeah, it is. Yeah, so the question is, do we get enrichment of PIP2 in the tube when we have, yes. So since the protein binds to PIP2, it turned out that it is enriching PIP2 as well. And if you don't have proteins, you don't enrich PIP2 in the tube. That's we have shown. Yeah. Not more than the rest. The PIP2 is not changing. I don't know. It's just the fact that somehow all together, so we don't describe the molecular, at the molecular level, this friction. It's a very phenomenological description that we have. And probably the fact that you immobilize lipid is important. I mean, the fact you have an amphipathic helix is important, but we don't have at all a molecular description of that. It's a phenomenological one. Yes. Marcos? Yes.
One conical one.
Yeah.
Conical one, which cylindrical one. I was expecting the cylindrical to actually feed, because it's flat, just one feed.
Yeah. So it's true that I mean, I didn't show the data. So it turned out that if to a certain extent, so you get in fact, you get something which is 1, and if you go to high curvature, you see a decrease. That's exactly what we see, which is again in agreement with the idea that if you have now if you have a stiff component to the membrane, it should be depleted at some point. Exactly. Yeah.
Okay. Shall we thank Patricia? Thank you.